So everybody is get joined here. मेरी आवाज आ रही है?
Yes, sir.
Yes, sir.
ओके, सबने ज्वाइन कर लिया है? कितने स्टूडेंट हैं आप लोग में से? हाउ मेनी नंबर ऑफ स्टूडेंट्स इन पीएचडी प्रोग्राम?
Sir, 5.
5 ही हैं. ओके, आई थिंक 3 आर गेट कनेक्टेड. So should I start?
Yes, sir.
Okay. So the today's topic for the PhD class is PCR. So I just go through the slide. So now it is get visualized to all of you.
Yes, sir.
Okay, fine. So PCR, a brief history of PCR. If you go through the brief history, then just some few points we have to just focus on it here in the history of PCR. First of all, you know the structure of DNA. DNA is a double helical structure, and if you see this double helical form, now this double helical form of the DNA is get arranged in a complex manner, like one helical turn, this, and finally it get converted into a more complex form as like a chromosome inside the cell. Okay, and DNA having the anti-parallel strand. One strand run from the 3' to 5' direction, and one another is done from the 5' to 3' direction. And the unwinding of the DNA is get occur with the help of the enzyme helicase. So how do we identify and detect a specific sequence in genome? So the big issue regarding that how to identify and detect a specific sequence in genome, there are a lot of other sequences in a genome that we do not interested in detecting, and that is the specificity problem. The amount of DNA in samples we are interested in very small. जिनकी हमें जरूरत है वो बहुत ही स्मॉल क्वांटिटी में होते हैं, और जिसमें हम नहीं इंटरेस्टेड होते हैं वो उसमें हमारे पास बहुत बड़ी संख्या होती है, बहुत ज्यादा सीक्वेंस होता है. तो ये हमारा एक बड़ा प्रॉब्लम है कि जो हमारे हमारे काम की जो चीज है, जो हमारे काम की जो सीक्वेंस. हेलो,
We were not
यस,
getting your voice.
नॉट
Now
गेटिंग,
audible. Yes, sir. Now audible.
नाउ ऑडिबल. Okay.
हमारे साथ ही तो नहीं है ये प्रॉब्लम।
So you are getting my voice? I am audible?
Yes, sir.
Okay. Now, so how do we identify and detect a specific sequence in genome? जो किसी भी सीक्वेंस या जिनोम में किसी सीक्वेंस को आइडेंटिफाई करने में जो मेजर प्रॉब्लम आता है कि जो हमारा खुद का जो डीएनए है जिसमें हम इंटरेस्टेड हैं उसकी क्वांटिटी बहुत ही स्मॉल होती है, सो दैट इज अ वेरी स्मॉल इन क्वांटिटी, और इसीलिए हमें उसको एम्पलीफिकेशन करना पड़ता है। जब हम उसके ऊपर काम करेंगे देन वी ट्राई टू बी एम्पलीफाई their DNA samples. So how do we identify and detect a specific sequence? So that similar question we have to move up, and you can see that हमारे पास एक huge sequence है। जैसे अभी हमने बात किया कि there's a lot of other sequence in a genome that we are not interested in detecting. तो बहुत सारे जो सीक्वेंस है जिनमें से हमें अपने काम की चीज निकालनी है, लेकिन जो बहुत सारा जो सीक्वेंस है सो वो उसको हम कैसे ओवरकम करेंगे। अब फॉर एग्जांपल आप देख रहे हो यहाँ पर हमने कुछ डेटा रखा हुआ है। उस डेटा में हम देख रहे हैं कि दीज ऑल आर द डेटा, लाइक पाइन में करीब करीब 68 बिलियन बेस पेयर्स होते हैं, कॉर्न में करीब 5 बिलियन बेस पेयर्स हैं, सोयाबीन में 1.1 बिलियन बेस पेयर, ह्यूमन में 3.4 बिलियन बेस पेयर्स, हाउस फ्लाई स्मॉल इंसेक्ट दैट इज 900 मिलियन बेस पेयर, राइस में 400 मिलियन बेस पेयर, HIV वायरस में 9.7 थाउजेंड बेस पेयर। सो यू कैन सी दैट कि कितना ह्यूज क्वांटिटी ऑफ genome is existing, the sequence is get existing in very very small creature of this life, and ultimately that DNA are। तो इसमें से हमें जो छोटे से सीक्वेंस जिस पे हमें काम करना है, हाउ वी कैन बी गो फॉर द दैट प्रोसेस। सो if it is just for the information कि अगर हमारा जो जीनोम है ह्यूमन जीनोम जिसकी जो टोटल नंबर ऑफ बेस पेयर इसमें 3.4 बिलियन है, इफ द बेसिस वे रिटन इन स्टैंडर्ड 10 पॉइंट, अगर हम इनको 10 पॉइंट में लिखने और इसको मेजर करें तो आप ये देख लो जैसे कि हमारे ये 10 पॉइंट्स है, 11 to 12 के बीच में ये हमारे 10 पॉइंट्स हैं। इन 10 पॉइंट्स के बीच में अगर हम इनको लिखना चाहें इन 3.4 बेस पेयर को, तो जो टेप होगा वो करीब करीब यही 10 पॉइंट्स में हम इनको 5366 माइल्स तक स्ट्रेच कर सकते हैं। तो दिस इज व्हाट यू कैन सी दैट कि इतना बड़ा जीनोम which may, if you can just sequence it into a 10-point scale, then it may arrange up to 5366 miles. Okay, identifying a 500 base pair sequence in genome would be like to finding that is a very problematic side, that how we can get this small sequence component from the huge stretch of these base pairs which is available inside the DNA. Now, how many molecules do we need to be able to see them? कितने मॉलिक्यूल चाहिए हमें इनको देखने के लिए? To be visible on an agarose gel, need around 10 nanogram DNA for fluorescent stain. अगर हम देख रहे हैं कि हमारे एम्पलीफिकेशन में अभी तक हमने जो प्रॉब्लम पहले देखा था स्पेसिफिसिटी की प्रॉब्लम थी, हम स्पेसिफिसिटी में ये सारी चीजें डिस्कस कर रहे थे। नाउ वी कैन सी द एम्पलीफिकेशन। जो फर्स्ट हमने स्लाइड देखा, हमने दो पार्ट यूज किया था, इसमें एक बिग इश्यू था स्पेसिफिसिटी की और दूसरा पार्ट था एम्पलीफिकेशन में। सो स्पेसिफिसिटी की सबसे जो प्रॉब्लम है वो प्रॉब्लम इज दैट इज बिकॉज ऑफ द ह्यूज साइज, ह्यूज बेस पेयर्स व्हिच इज अवेलेबल इन द डिफरेंट स्पीसीज। नाउ वी कम टू द एम्पलीफिकेशन। और जब हम एम्पलीफिकेशन में आते हैं तो हम किसी भी डीएनए को बहुत ही कम क्वांटिटी में भी हम इसको देख सकते हैं। लाइक अगर हमें फ्लोरेसेंट स्टेन के थ्रू डीएनए को अगर हमें विजिबल करना है तो 10 नैनोग्राम इज सफिशिएंट। 500 बेस पेयर प्रोडक्ट बैंड, it is approx this much mole of the DNA we can be get seen through the agarose gel. Okay, so now PCR solve both of these issues. So how do we identify and detect specific sequence in a genome? और टू बिग इश्यू की लॉट ऑफ जो सीक्वेंस है जीनोम में से जिसमें से हम किसी में इंटरेस्टेड नहीं है, और जो स्मॉल क्वांटिटी है जिस पे हम इंटरेस्टेड हैं, हम इनको कैसे सॉल्व कर सकते हैं। चाहे वो स्पेसिफिसिटी से रिलेटेड हो, चाहे एम्पलीफिकेशन से. The only component which can solve this problem is PCR. And what are the other very important functions of this PCR are? PCR helps in the forensic DNA detection. It identifies transgenic plants. It helps in the detection and quantification of viral infection. It helps in the cloning, helps in the detection of ancient DNA, and also helps in the gene expression analysis. So these all are the various methods for which we use the PCR. Okay, so you can see that forensic DNA के लिए you can see the crime investigations. We have to have a very common method of DNA profiling is one of the very common procedure. Transgenic plant को identify करने के लिए, which is not from the wild crop. If you have a transgenic plant and some other wild crops are there, and you go through the some change, see the variation in both of them, then that only the DNA profiling help you to identify that this transgenic plant. वायरल इन्फेक्शन, अभी आपने पेंडेमिक के टाइम में भी देखा, मोस्ट ऑफ द वायरल इज only be detected because of the that PCR methods. So these all are the various method. Nowadays it is a very commonly utilized, and this is one of the very good applications of the PCR. Now we come to the history. So you know the well-known name Carey Mullis. Okay, in what has been called by some of the greatest achievement of the modern molecular biology, Carey Mullis developed the polymerase chain reaction in 1983. Okay, तो ये बहुत पुरानी भी बात नहीं है. 1983 में कैरी मुलिस ने पीसीआर को डेवलप किया. PCR allow the rapid synthesis of designated fragments of DNA. Using the technique, over 1 billion copies can be synthesized in a matter of hours. Okay, so this is the one of the very good technique in which you, what you have to do, you have just getting the fragment of the DNA, and you can generate the million of the copies in a just hour, and you can see that huge quantity of the DNA you are having in your hand. And this overall technique is developed by this personality, that is Carey B. Mullis. PCR is valuable to scientists by assisting gene mapping, the study of gene function, cell identification, and to forensic scientists in criminal identification. Citus Corporation, Mullis' employer at the time of his discovery, was the first to commercialize the PCR process. In 1991, Citus sold the PCR patent to Hoffman La Roche for a price of $300 million. So you can see that it is not some government agency who can just initiate this process of the PCR development. It is the one of the companies, Citus Corporation, जिसमें मुलिस एम्प्लॉयर थे और उन्होंने वहीं पर अपना एक पीसीआर जनरेट किया, मेथड को जनरेट किया, और बाद में इसके पेटेंट को सिटस ने हॉफमैन रोज को बेच दिया at the amount of this much, $300 million. You can see that this is a huge amount. On that time it is get sailed up. Okay, now the inventor of the DNA synthesis process known as the polymerase chain reaction, the process is an invaluable tool to today molecular biologists and the biotechnology corporations. So this is the one of the note which is just seen here in 1944. Sorry, it is just a period of then after the 1983 that it is get seen that PCR is the process of invaluable tool to today's molecular biologist and the biotechnological corporation. Now, if we just see about the Mullis born in Lenoir, North Carolina, attending the University of Georgia Tech for his undergraduate work in chemistry, and then obtained a PhD degree in biochemistry from Berkeley. So, Mullis की brief history थी. In 1983, working for Citus Corporation, Mullis developed the polymerase chain reaction, a technique for the rapid synthesis of the DNA sequence. The simple process involves heating a vial containing the DNA fragment to split the two strand of DNA molecule, adding oligonucleotide primers to bring about reproduction, and finally using polymerase to replicate the DNA strand. Each cycle double the amount of DNA, so multiple cycle increase the amount of DNA exponentially and creating a huge number of copies of the DNA fragment. Okay, so this is what the PCR is. You have already, I think, you are get aware about the what the PCR reaction, but you can see that initial time when the Carey-Mullis is get to go through this technique, it is a totally a newer thing for each and every molecular biologist and the biotechnological companies, that this is the method through which revolutionized the so many work in the field of this molecular biology and the biotechnology. Okay. Now, the process which Dr. Mullis conceptualized in 1983 is hailed as one of the monumental scientific technique for the 20th century, a method of amplifying DNA. PCR multiplies a single microscopic strand of the genetic material billions of times within hour. Okay, so it is a method of amplifying DNA, and PCR multiplies a single microscopic strand of genetic material billions of times within hours. And what the Mullis explained, Mullis अपने terms में इसको किस form में explain करते हैं? It was a chemical procedure that would make the structure of the molecules of our genes as easy to see as billboards in the desert, and as easy to manipulate as tinker toys. It would find infectious disease by detecting the genes of pathogens that have difficult or impossible to culture. The field of molecular paleobiology would blossom because of PCR. Its practitioners would inquire into the specify of evolution from the DNA in ancient specimens, and the DNA was finally found on other planets. It would be a PCR that would tell us whether we had been there before or not. So these all are the advancement which is just done because of the PCR, and Mullis itself declared that it is the wonderful discovery at that time, and now we have to go through that complete process of the PCR, how the process is get occur, and how the PCR helps into the in various molecular biology aspects. Take all the MVPs from the professional baseball. Okay, so that is throw a dozen favorite movie star and half dozen rock stars for good measure. Add all the television anchor people now on the air, and collectively we have not affected the current good or the future welfare of mankind as much as Carey Mullis. That is the term which is used by for the Carey Mullis by Ted Koppel on ABC Nightline. This is the magazine in which they can just make this concept about the PCR, which is invented by the Carey Mullis. Now, what are the uses of the PCR? So, the first is use of the PCR in forensics. The PCR ability to amplify even the smallest amount of DNA from samples collected at crime scene, given the method, great power when used in criminal forensics. The DNA from bloody fluid, hair, or other tissue sample is amplified to create a nearly unique pattern for each individual. This pattern can be compared to suspect in the case. The infamous O.J. Simpson case was the first one in which the technique of PCR being widely publicized. Okay, so you can see that okay, because of the PCR involved in the forensic, we can investigate the so many criminal cases with the help of the PCR method. और इसमें हमें केवल और केवल बहुत छोटा पार्ट डीएनए का चाहिए होता है किसी सस्पेक्ट के बॉडी फ्लूइड से, हेयर से या किसी भी टिश्यू सैंपल से. हम उसको एम्पलीफाइड करके उसके यूनिक पैटर्न के साथ इंडिविजुअल को मैच कराकर we just what we do, we can see the either the suspect is really involved in that crime or not. सबसे फर्स्ट पहली बार एक फेमस केस ओजी सिम्पसन केस के लिए पीसीआर का यूज किया गया था। जीएमओ फूड डिटेक्शन के लिए, जेनेटिकली मॉडिफाइड फूड्स, इनके डिटेक्शन के लिए भी हम पीसीआर को यूज कर रहे हैं। वी नो दैट कि जो जेनेटिकली मॉडिफाइड फूड्स हैं वो यूएस में बहुत ही वाइडली ग्रो किए जा रहे हैं। सो the country which are not be in favor of the GMO foods, what they want to be that none of the GMO food or their seed and other component come to their own country. So what they do, whosoever we bring any type of the seed component or the plant component from the other one country to another, first of all they check it either that food is from the GMO variant or they are the from the wild varieties. So for that they go for the PCR testing. Okay, so for various reasons some countries require exporters to indicate the percentage of GMO content in grain and the food shipment, and that percentage is always be get checked in each and every shipment. PCR can be used to accurately measure the exact quantity of genetically modified food in a shipment by looking at the DNA that make up the food. Now, it is another very important area in which the PCR helps a lot, that is paternity testing. PCR power and identifying individual genetic makeup has made it invaluable for use of paternity testing. By amplifying specific DNA fragments from parents or close relatives, it is possible to reconstruct relatedness between individuals. PCR can only identify relationship between people today, but can also be used to identify historical family relationship. Okay, so in this way, you can see by just having the DNA fragment from the parents or close relatives, and we can identify the relationship between the people and also identify the historical family relationship. There is so many issues. You can just go through the magazine and the newspaper, then you have find out the so many paternity related issues are there, which is just nowadays gets simply solved because of the this PCR method, that is a paternity test. Archaeological, here is another very important area in which the PCR is get used. PCR has been used for many scientific studies in the field of archaeology, reconstructing of Dead Sea Scrolls, identifying of paint pigments in cave paintings, determining relatedness between individual in ancient ossuaries, constructing dinosaur from blood preserved in amber specimen. These all are what that is the archaeological areas where we can see the uses of the PCR. Now, PCR is also is the one of the most important area where the PCR help, that is the disease diagnosis. In this pandemic, you can see that how the PCR helps the identifying the viral infection inside the human individual. So PCR is now available in modern disease diagnosis. PCR can identify disease causing organism much earlier than other method, since it looks for the DNA of the organism itself, not its protein or its effect in our immune system. PCR has even been used to diagnose disease of the past by amplifying minute amount of disease-related DNA in preserved specimen. Okay, so you can see that PCR can identify disease-causing organisms, and PCR has even been used to diagnose disease in the of the past by amplifying minute amount of disease-related DNA in preserved specimen. So they can be just diagnosed in both the way. हम किसी हिस्ट्री के बेस हिस्ट्री को बेस लेकर भी डिजीज डायग्नोसिस कर सकते हैं, या कोई नया डिजीज भी अगर आ गया है तो भी हम उस डिजीज के डायग्नोसिस विद द हेल्प ऑफ पीसीआर वी कैन बी एबल टू डू दैट. So in the both the method we can be use the disease diagnosis with the help of the PCR. In the treatment also, PCR can not only use in the disease diagnosis but also in the treatment of the disease. For example, real-time PCR is used to directly monitor the amount of HIV virus in patient suffering from infection. By monitoring the amount of virus present, the drug therapies can be continually adjusted to maximize virus suppression. So this is how they can be help in the disease treatment also. Just by seeing the infection of any virus or the bacteria or any other, we can just maximize the drugs or minimize the drug, and through this we can be simply get the disease treatment also. Wildlife conservation. PCR can be used to identify not only individual but also can differentiate between species. It is often used in the wildlife conservation research. PCR can be used to monitor trade in products made from the endangered species. PCR can be used to monitor ecosystem for the presence of certain species. PCR can be used even to monitor and identify individual animals. So you can see that how many beneficial role of the PCR is there. Okay, as a pharmaceutical student, I think you have a get more focused on the PCR for the disease treatment or the disease diagnosis. But environmentally, you can see either in archaeological, in the wildlife conservation, PCR helps a lot in so many areas. In basic research also, like you can well known about the Human Genome Project. This project is only be completed because of the PCR. If there is no PCR method, then I think we are unable to get the completed with the Human Genome Project. So the Human Genome Project has identified tens of thousands of genes in a human genome. A key question is, what do these genes do? Part of the answer comes from the determining when the genes are turned on and off and what affect the level of gene expression. Quantitatively, PCR is a key component of determining the level of gene expression, and it is a critical tool in cancer research, disease studies, and development biology. So you can see that not only in the Human Genome Project, it can help in the identifying the thousand of the genes in the genome, but also it may help in the giving the answer, the type of the gene turned on and off, level of the gene expression, determining the level of the gene expression, and also it gives the some tools in cancer research, disease study, and the development biology. So these all are the component which where we can use the PCR lot. Now we come to the next topic, that how PCR works. So for the PCR, what we do, we have required the some very basic component. Here we go for the one animation which is developed by the BioRed company. Okay, we see the animation and see how the PCR get work. So here is one important component. We see that in the PCR, what is required? Here we require a DNA. DNA unwinding is get occur, and that unwinding is further get the polymerase enzymes is get binded to that unwinded strand, and after that, when we give the tag polymerase, then there is a that is initiation of the newer band synthesis get occur. So here we see this. So this is original template. What are the component? Primer is required. Now you can see that there is a separation of the strand is get occur. Primers are get binded to the single strand part. Further on, the enzyme is get binded and the reaction is get completed. So that you can see that different cycles is get see. ये cycle 2 में है जो हमारा similar process goes on. Cycle 3, that further strand are separated. Now the attachment, then polymerization, and finally that more strands are get formed. Now the copies are get multiplied and multiplied in each and every cycle. Cycle 6, 64, cycle 7, 128, cycle 8, 256. So in this way, as the Process where goes on. Now the overall synthesis is get occur, and this is what the how the PCR is get work on. Okay, one more time you can see that animation. सेपरेशन हुआ, सेपरेशन के प्राइमर बाइंड हुआ, फिर हमारा एंजाइम की बाइंडिंग एंड फाइनली सिंथेसिस. ओके, पहली साइकिल कंप्लीट हुई, दूसरी साइकिल में फिर मेल्टिंग, एनिलिंग, पॉलीमराइजेशन. थर्ड साइकिल melting, annealing, polymerization. Fourth cycle melting, annealing, polymerization. So in this way, the overall process of the this reaction, PCR reaction is goes on. Now the first step you can see that denaturation, anneal, replication. Second cycle, then further denaturation, annealing, replication. Third cycle, that is annealing, replication. Fourth cycle. So this is how the cycles, as the cycle is get increased, you can get the multiple number of the copies of the PCR reactions. So what are the reaction components are? So reaction components are water, buffer, DNA template, primers, nucleotides, magnesium ion, and DNA polymerase. So, if you want to perform the PCR in the lab, then these are the components you required. You required water, you required buffer, you required DNA template in which you are interested, then some primers, nucleotides, magnesium ions, and polymerase, DNA polymerase enzyme. Water. Why water? Water is the medium for all other components. So water is necessary because it is the one of the medium in which all over the processes get occur. That's why we require the water. Buffer. Buffer helps in the stabilization of the DNA, polymerase, DNA, and the nucleotides. So all of the component like DNA polymerase, enzyme, DNA template, and the nucleotides are get stabilized in buffer. And how we can create a buffer? Either we creating with the this KCl, Trish HCl, pH up to 8.3, or we can also add the tritan or twin component in the buffer. Okay, so by these component we can prepare the buffer. It is KCl, Trish HCl at the of the pH 18.3, concentration 100 millimolar, and the triton or twin is get used for buffer. DNA template contain region to be amplified, any DNA desired, purity not required, should be free from polymerase inhibitor. तो जो भी डीएनए टेम्पलेट होगा हमें इस बात का बहुत ही ज्यादा ध्यान रखना होगा कि वो पॉलीमरेज इनहिबिटर से फ्री हो, बिकॉज बहुत सारे डीएनए टेम्पलेट के साथ में ही हमारे क्या होते हैं, पॉलीमरेज इनहिबिटर्स भी होते हैं. सो we always we try to be there. There's no any type of the polymerase inhibitors are there. Otherwise, DNA polymerase enzyme not be get work on, and your not amplification is not be done. Primers. Primers are what? They are the specific for end of amplified region. So it is another very important thing about the PCR reaction that we have to see the primer which actually bind either the 3' end or the 5' end. And for this primer, it is most important that if you want to do the PCR of unknown sequence, you can add your unknown sequence in between the some known sequence, and then after you can go for the PCR, because if you have a known sequence, then there is a that the primers are get attached to the part of those known sequence, otherwise not. And if there's unknown sequence, you are unable to get any type of the proper specificity of the region, so that not the proper binding is get occur. So we took the forward and the reverse, both the primers in the PCR. And annealing temperature should be known, or how you can know the annealing temperature depending upon the primer length or especially the GC content. So both of these two help in the that we get the annealing temperature. Length is approx 15 to 30 nucleotide, or concentration is 0.1 to 1 micromolar. So this is what for the some primers, this is the one of the things which you have to consider in the primer, कि पहली चीज है कि उनकी स्पेसिफिसिटी होना चाहिए एम्पलीफाइड रीजन के साथ, फॉरवर्ड और रिवर्स प्राइमर होने चाहिए, एनेलिंग टेम्परेचर आपको पता होना चाहिए कि, और एनेलिंग टेम्परेचर किसके ऊपर डिपेंड करेगा, जीसी कंटेंट पे, कितने अमाउंट पे जीसी कंटेंट है उसके ऊपर ये एनेलिंग टेम्परेचर डिपेंड करता है। लेंथ करीब 15 से 30 न्यूक्लियोटाइड का हो सकता है, और कंसंट्रेशन 0.1 से लेकर 1 माइक्रोमोल तक we have to put it at the concentration of the primers. Nucleotides, you know, that is very important for the adding the nucleotide, adding the growing chain, that is activated NTPs, जिसमें DATP, DGTP, DCTP, DTTP. So these are the component which are the part of the nucleotides, and that should be always be there with the these nucleotide components always be there with the that overall components. If there is no NTPs, so NTPs, then I think no lengthening of the chain is get occur. Another very important component about the PCR reaction is that is magnesium ion. So essential cofactor of the DNA polymerase, magnesium ion is essential cofactor of the DNA polymerase. It requires in a too little amount. An enzyme would not work without the magnesium ion. It stabilizes the DNA double helix. Too much, if it is a very high in amount, then it causes DNA extra stable, non-specific priming and bad smearing. So बहुत कम भी नहीं होना चाहिए, बहुत ज़्यादा भी नहीं होना चाहिए, बहुत कम हो जाएगा तो एंजाइम काम नहीं करेंगे, और अगर बहुत ज़्यादा हो जाएगा तो डीएनए जो है वो एक्स्ट्रा स्टेबल हो जाएगा, नॉन स्पेसिफिक प्राइमिंग होगी, और बैंड स्मेयरिंग जो है वो बहुत ज़्यादा होगा। और इसकी कंसंट्रेशन कितना चाहिए, लगभग 0.5 to 3.5 माइक्रोमोल इन द एसिड. दिस इज द अमाउंट व्हिच इज रिक्वायर्ड फॉर द magnesium ion. And the most important component is the DNA polymerase, the enzyme that does the extension, tag or similar, heat stable होना चाहिए, जिसकी concentration approximately 1 unit per reaction. This is what that requirement of the DNA polymerase. Two more major things, that is it should be a heat stable, and the approx concentration is approx 1 unit per reaction. So the summary: the water, buffer, DNA template, primers, nucleotide, magnesium ion, and the DNA polymerase is the overall reaction component of PCR. Setting up PCR reaction. So this is what we go for the some typical PCR reaction to be get set up. If you want to be a total volume of 50 microliter, then we can be use this overall content. Sterile water करीब 38 microliter, PCR buffer 5 microliter, magnesium ion that is 2.5 microliter, DNTP is 1 microliter, primer forward and reverse both 1 microliter, DNA polymerase 0.5 microliter, and DNA template should be of 1 microliter, and this overall give the 50 microliter of the concentration. सो मिक्सिंग कॉमन रीजेंट, अगर हमने कुछ कॉमन रीजेंट को मिक्स किया तो हम उसको ये हमारे समय बचाते हैं. लाइक अगर हमने यही सेम कंपोनेंट को अगर हमने 20x पे बना लिया, दैट इज 980 माइक्रोलीटर, तो उसमें से अगर हम 1x यूज करें तो दैट गिव द सिमिलर कंसंट्रेशन. So there is no requirement of for each and every step you can be required the addition of all of these component. So what you have to do, in the safer side, you go for the preparation of 20x these PCR mixtures, and ultimately from this you can create a 1x and use it as such. Okay. So this is how another very important thing about the master mix. ये हमारे पास ऑलरेडी रेडी टू यूज आते हैं, हमें कुछ नहीं करना है, ऑलरेडी इट इज अवेलेबल मास्टर मिक्स. इसमें what we add, only we add the few things, like we have to add uh, that is reverse and forward primers and some DNA template, and other rest of the component are totally available in the master mix. So sterile water, 10x buffer, MgCl2, DNTPs and DNA polymerase are already available in the master mix, and इसकी concentration करीब 19.6 microliter, you if you can use it, and rest of the component you use the DNA template and the that forward and reverse primer, and your purpose is get solved, and you can directly go for the PCR reaction. Programming the thermal cycler. If you have already the PCR machine, so there these are the some steps you can be follow. Like you can go for the initial denaturation up to 95 degrees centigrade for 3 minutes, DNA denaturation 95 degrees centigrade up for 1 minute, primer annealing that is for 65 degrees centigrade for 1 minute, primer extension up to 72 degrees centigrade for 1 minute, and go to step 2, that is for repeat the cycle for 39 more times, and finally the process is get in. So here the primer annealing, you can be go for the more standard process for the primer annealing also, just by calculating the GC content of your primer. And by if you have know that the content are not be such varied, so you can be use this primary annealing temperature as such. There's no some any other information is required for the primer annealing. But if you didn't sure about your primer, about what the amount of the GC contents are there, so you are first of all go for the that calculation of the GC content, and then after you go for this primer annealing temperature. Analyzing the amplified DNA. So for if you can simply complete your process of the PCR, then how you can visualize. So after thermal cycling, tubes are taken out from the PCR machine. Content of tubes are loaded onto a agarose gel. DNA is separated by size using an electric field. DNA is then stained, and PCR products are visible as different bands. So you can see this is a PCR machine. After completion of the cycle, you can just took out the tubes, add your that separated bands into the separated DNA into the that wells you have created on the agarose gel, and finally run the agarose, run this electric, start the electric field, and finally your bands are get separated according to the size, and this is how you can see the bands are get visualized on agarose gel. This is what you can see that it is an electrophoretic apparatus. Here the agarose gel is get assembled here, and the wells are get created, and your isolate your amplified DNA are get inserted here in this well, and finally you can be see that negative to positive the electric field is get run, and finally your DNA bands are get separated out. So you can see in this picture this is a gel running, and finally the bands are get separated, and it is get visualized. So after the gel has run, it is stained to reveal the DNA band. So अगर जेल हमारा रन कर जाता है तो हमें ब्लैंक सा ही दिखता है, कुछ दिखता नहीं है, बट जैसे ही हम उनके बैंड सेपरेट हो जाते हैं, then we have to get visualized in this way. The bands are get separated. The final result of the traditional PCR procedure is a gel with a series of bands. यहाँ marker add करते हैं, and rest you can see that are the band. Marker give the size of the band. Here, whatever the size are there, it is get only be get predicted with the help of this marker. So band can be compared against each other with the known size standard to determine the presence or absence of a specific amplification product. So that's all about the complete lecture. Thank you. So now if you have any query, then just you have to get asked for PCR. Yeah. Anybody have any question regarding the PCR? Because today we have discussed in detail about what are the different application of the PCR, where these PCR are get used, what is the actual procedure, how we can perform the PCR, what are the components which are used in the PCR, and how the machine is get set up for the PCR reaction. After the completion of the PCR process, how we can be simply check the our PCR products. Okay, so that's all about the complete PCR. If you have any question regarding this the today's lecture, just you can ask if you have.
Sir, what is the GC content as you shown in slide?
Yeah, GC content. आपने डीएनए की कंप्लीट स्ट्रक्चर को पढ़ाई होगा, डीएनए पढ़ा है
Yes,
ना?
sir. Yes, sir.
तो डीएनए में आप देखते हो कि हमारे पास 4 बेसिस है. ओके, कौन कौन से बेसिस हैं? ए टी जी सी.
GC
ओके, ए जो है वो हाइड्रोजन बॉन्ड किसके साथ बनाता है? टी के साथ.
के साथ में. Yes, sir.
ओके, ए का जो हाइड्रोजन बॉन्ड बनता है वो टी के साथ बनता है और उसके बीच में 2 हाइड्रोजन बॉन्ड होते हैं.
Okay.
लेकिन जी का जो बॉन्ड बनता है वो सी के साथ बनता है, 3 हाइड्रोजन बॉन्ड बनते हैं उसमें.
Yes, sir. Yes, sir.
ओके, तो जितना ज्यादा हाइड्रोजन बॉन्डिंग होगी उतना ज्यादा टेम्परेचर चाहिए उस हाइड्रोजन बॉन्ड को ब्रेक करने के लिए. ओके,
Okay.
तो
Okay.
जहां पर जीसी कंटेंट ज्यादा होगा, इसका मतलब है कि वहां डीएनए स्ट्रैंड में हाइड्रोजन बॉन्ड की कंसंट्रेशन ज्यादा हो गई. तो हाइड्रोजन बॉन्ड का कंसंट्रेशन ज्यादा होगा तो टेम्परेचर भी ज्यादा चाहिए होगा उन डीएनए को अनवाइंड करने के लिए, सेपरेट करने के लिए. ओके, इसलिए हम जीसी कंटेंट को जरूर चेक करते हैं जब हमारा एनिलिंग टेम्परेचर हम सेटअप करते हैं. अगर हमारा एनिलिंग टेम्परेचर हमें ऑलरेडी पता है कि इतने पर सारे सभी तरह के हाइड्रोजन बॉन्ड इजिली ब्रेक हो जाते हैं और हमारा डीएनए स्ट्रैंड सेपरेट हो जाता है, तब हमें जरूरत नहीं होती. बट लेकिन अगर किसी कोई ऐसा प्राइमर सेटअप है जिसमें हमारा जीसी कंटेंट बहुत ज्यादा है, तो हमारा ऑब्वियस सी बात है कि हमारा हाइड्रोजन बॉन्ड का कंसंट्रेशन ज्यादा हो गया. तो हाइड्रोजन बॉन्ड का कंसंट्रेशन ज्यादा होगा तो हमें थोड़ा और टेम्परेचर देना पड़ेगा डीएनए को अनवाइंड करने के लिए. ओके, और साथ में एनिलिंग टेम्परेचर. एनिलिंग टेम्परेचर क्या? अटैचमेंट के लिए. तो हमें अटैचमेंट कराना है अगर, तो हाइड्रोजन बॉन्ड के लिए 3 बॉन्ड चाहिए ना, 3 बॉन्ड के लिए टेम्परेचर हमें फिर ज्यादा चाहिए टू गेट द अप्रोप्रिएट. तो दिस इज व्हाट द सिमिलरली कि फॉर द ब्रेकिंग और फॉर द रीअरेंजिंग और अटैचिंग द that DNA strand with the primer, we require the good amount of energy. That's why we have to get the calculation of the GC content. Okay, तो यही है GC content, वही है
Okay,
हाइड्रोजन
sir.
बॉन्ड का
Thank you,
नंबर.
sir.
Anything else? Okay, I think that's all for the today. So in the next turn we discuss more about some other techniques which is already get mentioned in your this topic. Okay, see you in the next class. Okay, now leave
Thank
you.
you, sir.
Okay.
Thank you, sir.